It's quite nice to go after Jason because he's already introduced quite a few acronyms and a lot of it jargon. Thank you so much, everyone, for being here. It's my pleasure to present a little update on Project MOSS, and MOSS stands for Marine Oil Spill Studies. If I can move forward the slide, here we go. I'm here as the project leader and spokesperson, but really this is a large team of GSC scientists, American scientists from academia as well, and there are communities, First Nations communities involved in the project. They've all contributed a lot of sample, ship time, expertise, gray matter, and I'm very, very grateful for them. In short, we are looking at the Kitimat Fjord system, Kitimat being a very active port on the north coast of British Columbia. It is most notably a gateway for the export of Canada's energy resources, these days mostly liquefied natural gas. There's also an aluminum smelter in Kitimat itself. And it's really a gateway to international markets. So what that means that there's a lot of ship traffic, a lot of tanker traffic in the area. The main objectives as a whole for the project are mostly, they're twofold. The first one is really to establish a baseline of natural variability in the system. And we're talking in terms of temperature, salinity, productivity, these kind of things, as well as assessing the current state of human impacts in the system. The second objective, the second main axis of the project, is about testing the capacity of microbial communities from the site to mitigate accidentally released oil. So in the event of an oil spill, we want to see if the natural environment has a decent resilience, and this is in light with the fact that the bottom waters are seasonally hypoxic, so there is oxygen limitation at depth in the entire system. Very broad strides. There are three major phases to this project. The first one, so we're heavily relying on sediment samples. This is our main tool to go out there and investigate current and past conditions. So we first have a kind of a calibration effort, and so using a set of surface sediment samples and sediment trap time series, we're calibrating the signals encoded by various biological and geochemical indicators. We're also using that set to assess current human impacts in the system. The next phase is about reconstructions, and these are ongoing. I'm not going to talk about them today, but we go back in time using sediment cores and using the same proxies that we've developed in the first phase. We go back in time and we reconstruct the past environmental conditions. So we have a couple of short box cores and we have two longer piston cores that cover pretty much the entire of the Holocene. And then the lab tests of microcosm experiments. This is how we quantify in the lab and how we measure the capacity of microbial communities to degrade spilled oil under a range of oxygen concentrations. So very briefly, this is a big system we're talking about. It's a big fjord system. It essentially acts as one big estuary, with the hydrological head being around Gardner Canal here to the east. This is where most of the fresh water enters the system. So it's an oversimplification to present it as one estuary, but it's essentially what it does. And so close to the head of the inlet here, we have Kitimat, the aluminum smelter is close to here, LNG Canada site is here as well. There are two rather small communities, I mean, in terms of potential human impacts. There's the Kitimat village, home of the Hassla Nation, about 700 inhabitants. Hartley Bay, home of the Gitgat Nation, about 200 inhabitants. And so really most of the human activities are concentrated towards the head of the inlet. So the entire system is seasonally hypoxic, and this is courtesy of a couple of seals at depth left behind by, so those are terminal moraines left behind by glaciers that retreated at the end of the last glacial maximum. And these hinder the bottom water ventilation. So thanks to colleagues at PGC and DFO, we got our hands on a large set, a really good set of surface sediment samples. We have some short cores, sediment trap, and longer sediment cores too. Quick view, there will be mostly teasers on preliminary results. So palynology is my main tool for paleo reconstruction. So palynology is the study of organic walled microfossils, and in the marine realm, we find a lot of things, but the most useful type of organisms are dinoflagellate cysts. So dinoflagellates live mostly in the upper water column, but in the life cycle, they produce resting cysts that we find in the sediments. And so by studying the sediments, we can study phytoplanktonic communities that choose to live at the time of deposition. So here you see a few examples. Alexandrium on the top left is never really good news. This is the cyst produced by Alexandrium, and when you have a bloom of Alexandrium, it causes paralytic shellfish poisoning. So it's never really good news, and everybody's interested in tracking it though, for obvious fisheries and shellfish harvesting in the area. So a lot of the autotrophic species, so cysts produced by autotrophic species that do photosynthesis themselves, they tend to have a clearer wall here, and you have quite a few examples. And here you also have more on the brown side, you have species that are produced by heterotrophic species. So these feed on other phytoplankton. This one, most notably Brigantodinium, it's the main player here. It's the most abundant taxon. It's a genus, and it feeds mostly on diatoms. A few other examples just to show you how pristine that system is. It's beautiful fauna, very diverse and very abundant dinoflagellate cysts in those samples. We also have a species that was very recently described as Botadinum pontifossatum with pontifossate ornaments. So this is my world when I look into the microscope. It becomes interesting when we map out those biological proxies across the system. So right now, this is preliminary data on the first few samples, but you can already see on the left-hand side, this is the dinoflagellate cyst to pollen and spores ratio, and it gives you pretty much a marine to terrestrial signal. And you can see the ratio is much higher in a more marine location and much smaller in more terrestrially influenced environments. Overall, the total cyst concentration, cysts seem to be pretty happy in those mid parts of the system. And this is the ratio of autotrophic to heterotrophic as well. I'm going to go quickly through that, but just to show you a couple examples of how different species respond differently to environmental factors. So this is our main player, and it seems to be very happy at that particular site, and it feeds mostly on diatom. So this is an indication of diatom productivity, high primary productivity. And on the other hand of the spectrum, we have the cyst of Archaeopterygium cyanici, and you can see it gets its highest concentrations in much more proximal stations. We did not just look at biological markers in that set of samples. So Jason had generated the data at Delta Lab, data on PAHs. And so those polycyclic aromatic hydrocarbons that we can use as traces of human activity. So here are two examples. On the left-hand side, this is rutene, and it's a marker used for conifer combustion. is pretty much close to background levels in most of the system, but you can see elevated concentrations close to kinemats, close to the major urban center. And then on the right-hand side, we have coronene, a marker used for fossil fuel combustion. And similarly, we see the greatest influence of human activity towards the head of the inlet, so the closer to the urban center. Now on to microcosm experiments. This was very exciting and completely new to me, so I had to pick a lot of brains. And again, this is really teamwork. So what we did, we gathered sediment and seawater close to the bottom on site. We put everything in bottles, and we added it's a type of fuel oil. It's the new standard for low sulfur fuel oil. So it's the fuel that tankers use as fuel. Natural gas itself is not really a problem for oil spills. If it spills, it will just sit at the surface and evaporate. But the fuel is more of a concern, especially to the communities on site. So that was our mixture: sediment, seawater, and fuel, and we're testing the natural capability of microbial communities to degrade that spilled oil under a range of oxygen concentrations. So we were forcing the oxygen concentrations in the headspace of each one of these bottles. We had our main setup, and then a no fuel control and a kill culture control. In short, because I don't have much time left, not much. So over the 4 months of the experiments, of course, nothing much happening in oxygen drawdown from the anoxic cultures. There's just no oxygen. But overall, we see a faster oxygen withdrawal in the oxic culture and slower from the hypoxic cultures, but ultimately similar results. And same as carbon dioxide production as a crude, rough indicator of microbial activities. Not much happening in the anoxic columns, and a faster response in the oxic conditions, but ultimately very similar, or if anything, the hypoxic conditions in the end were more productive. So we tracked the evolution of microbial communities throughout this experiment, and this is work from Casey Hubbard, his team at U of C. So roughly DNA yields as a rough estimate of microbial biomass constantly lower in anoxic settings, and then all trends were to increase the biomass at first and then drop. And then just to give you a little teaser as well, because there's a lot of data generated here, when we sequence each in detail the microbial communities, this is a phylum of bacteria that are known as petroleum degraders, and we can see an increase in hypoxic and oxic conditions of this particular genus of pH degraders. We do not see this increase in anoxic settings, and we do not see, obviously, this increase in our no fuel control. So when we add the oil, we do see the increase in these pH degraders, and same thing for those alkane degraders here. So just a little teaser. I'll wrap up with the petroleomics. So we also kept track of which components of the oils were being degraded. So this is work from Rachel Robinson and Andy Mort here at JC Calgary. In terms of N-alkanes, so the chains, typically the most easily degraded fraction across the 3 different oxygen settings. What you're looking at are spectra here, and the different time points are represented as curves. So the easiest is to start on the right-hand side. In the anoxic setting, we basically only have initial versus final. And as you can see, there isn't much change. Not much has been degraded over the 4 months of the experiments. But in the oxic setting, right away, we see a big change. So degradation of N alkanes happening quite fast. And hypoxic in the hypoxic experiments, it was a more gradual change, but ultimately quite similar. And then for PAHs, in this case, the entire time series is presented as those groups of columns here. And in the oxic, each bundle is a class of PAHs, basically. And so in the oxic and hypoxic environments, we can see a decrease in about 2 orders of magnitude from the beginning of the experiment to the end of the experiment, and not much happening under anoxic conditions. So these are just teasers. We're still working on generating some of the data and writing everything up. But it's pretty exciting so far. I think I run out of time, so I'll let people read that, but I'll be more than happy to answer any question you may have. Thank you.